So hi, I'm Ethan Chiu. So a bit about me. So I'm a first year Han research scholar at Yale. Went to Syosset High School, and I'm a U.S. presidential scholar and a Regeneron STS finalist. As for some of the activities I've participated in, I serve on the American Red Cross National Youth Council, coordinating events throughout the U.S. Related to things such as biomedical services with the Red Cross, the seat of next gen diplomacy. I did research at Mount Sinai. I'm American College of Cardiology Young, so doing research with cardiologists. And I was the former U.S. Youth Advisory Council for the UN Ocean Decade Policy Committee Chair. And as for my future goals, I'm hoping to work towards MD-JD or MD-NPH, working in healthcare research and policy. So as for the background of my projects, the world spends so much money in prescription drugs, and it's only expected to increase every year by exponential amounts, exceeding inflation. And global spending on cancer treatment specifically was already 150 billion in 2018, and for the last 5 years, it's been rising by 10% every year. And as such, so many budgets are becoming threatened. So many less people are getting access to treatments, prescription drugs that they need due to the costs. So pursuing novel uses for existing generics can actually reduce the cost of cancer treatments. And that's what I was focusing on. So to start off, I sought to create like more economical treatments for specific cancer called uveal melanoma, which is the most common eye cancer with an 80% mortality rate and a median survival of less than 6 months. So it's extremely deadly. And just looking at some of the studies that prompted this idea, tumor cells were thought to only use glycolysis, but recent studies have suggested that mitochondrial oxidative phosphorylation is also required for tumor growth. And also, given its mitochondrial oxidative phosphorylation inhibition, in previous studies, doxycycline seems to be effective, efficient, and economical to treat uveal melanoma, considering how expensive some of the other treatments are. And you can see uveal melanoma on the right. So currently, a lot of researchers use like animal and human cell lines to test treatments, but a lot of the differences between animal and human tissue prompt different results. And human tissues have a lot of different contaminations and mutations specific to individuals. So I was using like a in vitro seeing a human eye organoid model created using differentiated stem cells. And you can see the eye developmental cycle and the process of creating the human eye, I guess, sample on the right. So as for some of the specific equipment used, single cell RNA transcription was used as a computational biology analysis, and lentiviruses were used to deliver gene modifications through CRISPR, and immunofluorescence is used to detect and quantify protein expression, and qPCR is used to detect and quantify gene expression by amplifying the RNA and then the DNA by converting it to cDNA. So this is the specific methodology. If you want to ask me further questions about it in the future, feel free to, but given the time constraints, I'll just skip past this. So you can see here are some of the gene markers used to identify specific clusters. So there's like different markers, genes that correspond with different regions of the eye, so used to sort of sort out in computational analysis. And as for the it's an richer human gene atlas prediction for the specific area of the eye that corresponds to. So if you take a look at this, it predicts with the significant p-values how likely by inputting the genes it is to be a specific region of the eye. And here is like the different clusterings for it and specific like gene expressions within each area that was labeled with the eye. And as for lentiviral transduction, did a lot of basically inserting viruses, lentiviruses, in order to deliver CRISPR-Cas9 transfection vectors and then immunofluorescence and qPCR to specify like the markers that were analyzed. And as for my results, what I found was that there were a lot of, like the same model accurately represented the human eye based on the many gene markers analyzed, oxidative phosphorylation. In terms of that, doxycycline is found to significantly inhibit oxidative phosphorylation while increasing tumor suppressor genes. Ki67, STRO1, and UCB5, those are proliferation markers specific to the neural crest where uveal melanoma originates. AUCB5 is a uveal melanoma marker if it becomes proliferating a lot. So that was analyzed through fluorescence. And doxycycline's effects on uveal melanoma was also found in terms of increasing the expression of tumor suppressor genes and also decreasing the mitochondrial function, so basically preventing uveal melanoma from being able to continue proliferating if it doesn't have energy. Immunofluorescence specific platings and images were taken. This is the qPCR results normalized to the control values. So you can see some of the data analysis that I was able to do. So all of this is log, so it means that it's base 10. It's not based on like normal percentages. So basically there is a significant difference by 10 times for each different line going up. So as for my conclusion, I found that doxycycline significantly increases the expression of tumor suppressor genes like BAP1, SOX10, and P53 with minimal cytotoxic side effects, given that after 2 days of treatment, doxycycline didn't really affect the total cell count, proliferation, neural crest presence, no neural crest proliferation. Uveal melanoma was induced in the novel knockdown P53 and BAP1 knockdown organoid. You can see that the increased Ki67 proliferation, oxidative phosphorylation, tumor growth, and uveal melanoma prognostic factor within the neural crest. Doxycycline-treated UM organoids have significantly less neural crest proliferation, significantly lower levels of proliferation, oxidative phosphorylation, UM prognostic factor, and tumor growth through angiogenesis within the neural crest, which indicate a significant inhibition of uveal melanoma. And the hypothesis for this is that since uveal melanoma cells need more energy, then doxycycline likely targets them more, which causes their mitochondrial oxidative phosphorylation inhibition to be significantly greater than that of doxycycline-treated wild-type cells. And overall, considering that uveal melanoma has a current mortality rate of 80% and 6-month median survival period, and currently available treatments expensive and ineffective, the prescription antibiotic doxycycline is a prime, effective, efficient, and economical therapeutic candidate for UM and can help many in both the US and developing countries access treatment to cancer, hopefully at a cheaper price in future studies that I hope to conduct. So feel free to ask me about some of the future research in the future, but for the sake of time, here are some of my references. And thank you, GHLC so much and Johns Hopkins for giving me the opportunity to present. And also thank you to my mentors for providing me guidance regarding specific equipment to use to test my hypothesis, how to use specific equipment types, and thank you to the New York Eye and Ear Foundation for supporting the study through a grant. So here's my contact information, and thank you so much for listening.